You know, when you hear the word sugar, what comes to mind? Probably sweetness, right? Energy, something you eat. But what if I told you that the exact same basic molecule that sweetens your coffee is also responsible for building the tallest trees or even the tough armor on an insect? We're about to dive into this hidden world because the secret architecture of sugars, well, it's way more incredible than you probably think. And that right there brings us to the core mystery we're tackling today. Think about it. A potato is basically a dense brick of energy. A tree, on the other hand, is a massive tower of structural strength. And yet, at their molecular core, they are both built from long, repeating chains of the very same sugar, glucose. So, how on earth is that possible? Well, the answer isn't about what they're made of, but how they're put together. It's all about the architecture. You can almost see it here. It's like a fork in the road. One path gives you this coiled spring, perfect for storing energy. The other path gives you these rigid, powerful fibers, perfect for building things. And the choice between these two completely different outcomes, it all boils down to one single, tiny decision at the molecular level. All right, so let's check out that blueprint. If we're going to understand how nature can be so clever, building such wildly different things from the same starting block, we've got to get a handle on the basic design rules. And really, it all comes down to just 3 key factors. First, what are the building blocks you're using? The sugar units themselves, the monomers. Second, how are you linking them together? The type of chemical bond is critical. And third, how much are you letting the chain branch out? That's it. Those 3 choices, that's the whole architectural plan right there. And this, right here, is the absolute game changer. It's the most important decision in the entire process, the kind of bond you use. Check it out. On the one hand, you have an alpha bond. This bond forces the chain to twist into a coil, like a spring. It's open, it's accessible, it's perfect for storing energy. But use a beta bond instead, and you get a completely straight, rigid chain. This shape is perfect for stacking, like lumber, to build strong structures. It's just wild. One tiny difference in a bond creates 2 totally different destinies. Okay, so let's follow that first path. What happens when nature goes with the alpha bond? Well, you end up with a molecule that's basically a perfectly designed fuel tank. In plants, that choice gives you starch. You can really think of it as the plant's pantry. It's actually a mix of 2 slightly different designs. You've got amylose, which is just a simple, long, unbranched coil of glucose. And then you've got amylopectin, which is also a coil, but it has branches sticking off every 20 or 30 units or so. The key is both use those easy-to-break alpha bonds, making them super simple for the plant to tap into when it needs a quick burst of energy. Now, animals like us, we need an even faster system, so we use something called glycogen. Think of it as amylopectin, but on steroids. It's way more branched, a new branch every 8 to 12 glucose units, and it's packed together much more tightly. So why is this so clever? Well, check this out. Every single one of those branch tips is like a little docking station for an enzyme. The more branches you have, the more places enzymes can work at the same time. And that means you can unleash a huge flood of glucose into your system almost instantly. It's the ultimate quick-release fuel source for when you need to move, and move fast. All right, let's switch gears and explore that other path. What happens when nature picks the beta bond instead? Now we leave the world of coiled-up energy behind and step into the world of straight, incredibly strong building materials. This choice gives us cellulose, which, by the way, is the most common organic compound on the entire planet. Because it's built with those beta bonds, every chain of glucose is perfectly straight. And because they're straight, they can line up perfectly next to each other, forming these huge sheets that are held together by thousands and thousands of hydrogen bonds. This is what creates microfibers with just incredible strength. It's literally what gives a tree its toughness. And fun fact, our digestive enzymes are built to snap alpha bonds, not beta bonds. That's why we can't digest cellulose. To us, it's just fiber. But nature doesn't just stop there. It gets even cooler. If you take that same blueprint for cellulose and make just one tiny little tweak to the glucose building block, just swapping out one chemical group for another, you get chitin. That one simple change makes the whole material more water resistant. And the result? You get the hard, reinforced armor you find in the exoskeletons of insects and crabs. It's just amazing. Another tiny change in the blueprint, another huge difference in what it can do. So up to now, we've mostly been talking about simple chains made of just one kind of sugar. But this is where the system gets really powerful. This is when nature starts mixing and matching different building blocks, or even attaching these sugar chains to totally different molecules.
So when you start building chains with two or more different kinds of sugars, you get something called, and it is a bit of a mouthful, a heteropolysaccharide. A great example is what bacteria use. Their cell walls are made of peptidoglycan, which alternates two different sugar units to create this super rigid cage-like structure. And in us, in animals, that squishy stuff between ourselves, it's built from something called Glycosaminoglycans, or GAGs for short, which are also long chains made of two repeating sugar units.
And if we zoom in on these GAGs, you can see how they build something seriously amazing. See, they're covered in negative charges. This has two incredible effects. First, all those negative charges repel each other, which forces the whole chain to straighten out into a stiff, rod-like shape. And second, those charges attract a massive cloud of water molecules all around them. The result is this thick, hydrated gel. It's the perfect material for lubricating our joints and for resisting compression and our cartilage. It's basically a shock-absorbing cushion made of sugar and water. But sugars are so much more than just fuel and building blocks. This is where they take on their most sophisticated role of all. When these sugar chains, they're called glycans, get attached to proteins or fats, they become information carriers. They basically act like complex antennas sticking off the surface of our cells, handling things like cell recognition, communication, and even identity. These hybrid molecules are called glycoconjugates, and there are three main types you should know about. First, you have proteoglycans. These are enormous molecules with a protein core that's just decorated with those long GAG chains we talked about. They make up the bulk of that matrix between our cells. Then you have glycoproteins. These are proteins with smaller, more complex branch sugar chains attached to them, and they are absolutely vital for cell signaling. And finally, glycolipids. These are fats in the cell membrane that have a sugar attached, and they act as cell surface markers. In fact, they're what determine your blood type. Okay, so let's zoom back out and just take in the big picture for a second. From coiled-up energy reserves to super strong structural beams to carriers of complex information, it all comes from one simple, incredibly elegant design system. And this really pulls it all together. You start with a simple sugar building block. The first, most critical choice is the bond. Go with the alpha bond and you get a coiled shape, perfect for storing energy like in starch or glycogen. Go with the beta bond and you get a straight, fibrous shape, perfect for building things like cellulose and chitin. And from that one choice, you can get even more complex. You can mix in different building blocks to create specialized gels and matrices, or you can attach them to proteins and fats to carry biological information. It's this whole amazing system of function that all flows from just a few simple architectural rules. And that right there, that's the heart of it all. It's this idea that a few fundamental architectural rules generate the vast functional diversity of the glycan world, from simple fuel to the complex language of the cell. It's just an unbelievably powerful and efficient system. Which really just leaves us with one last thought to chew on. If changing a single chemical bond can be the difference between a potato and a tree, between food you can eat and fiber you can't, just what other incredible secrets are hiding in plain sight, coded into the simple architecture of the molecules that build our entire world?